you can look here at these references later. The very first one on the archive is a recent review article, so it's accessible to non-specialists. First, let me talk about advances in the use of AI and ML to predict phenotype from genotype. So this is something that's happened just in the last few years as we have up to millions of genomes and good algorithms to analyze them. We can now identify high-risk individuals by genotype for most important disease conditions. So in the graph on the right, the age is on the horizontal axis, and people classified as red are high risk according to DNA information alone, and dark blue, low risk according to DNA information alone. And you can see there's a dramatic difference in the probability in the course of life that a high or low risk individual will have the condition. I'd like to comment that the number of women who, through polygenic risk scores, can be identified as high risk for breast cancer is about 10 times larger than the number of women who carry the BRCA variant. So it's a much larger population of women that are walking around not knowing they're high risk for breast cancer, but we can easily identify them now. Here's another set of results. The polygenic scores on the horizontal axis, and the nonlinear curve is showing you that as you go into the tail, if you look at people with high or very low polygenic scores, the absolute probability that they will have the condition in their lives goes up in a very nonlinear way. And so for some traits like hypothyroidism, the indicator on the right, we can already say at birth, if you just give us a sample of DNA from a baby, that maybe with probability 0.8, that baby, when she or he is 40 or 50 years old, is going to have hypothyroidism. So it's a rather dramatic advance in our ability to map genotype to phenotype. Here's another predictor. We can predict the height of an individual with error about 1 inch. On the left-hand graph, the scatter plot, the blue dots are men, the red dots are women, and the little horizontal black lines are the standard errors. And so most people are within a few centimeters of what is predicted just from DNA alone. And on the right, you can see the horizontal axis is the position if you were to lay out your 23 chromosomes end to end. And so there would be about 3 billion base pairs there. The individual lines there are indicating specific variants that are used in height prediction that seem to affect people's height. There are about 20,000 of those variants. So it seems like a very complex trait. It is one of the most complex traits that has been solved. However, as a fraction of the total number of features, there are millions of features, actually billions of potential features, which could influence height. And so all of these predictors, although they're highly complex, they are, in the machine learning terminology, sparse. They only use a very small fraction of the possible features that are contained in the information in your DNA. So what we're really talking about here is really very fundamental advances in human genetics coming from AI and data. And as I keep emphasizing, this is really the ability to map genotype to phenotype, to fully decode the genome and to tell from someone's genome how they're different from other individual humans. And so there are many examples now of quantitative traits and also disease risks that we can predict fairly well. I want to mention a few things about genetic architectures in general that we've just learned now in the last few years. So first of all, surprisingly, at least to some people, a very large part of the control knobs for these predictors reside in non-protein coding regions. So it's not just the genes which code for individual proteins or RNAs that are important here. For some predictors, 80–90% of the variance in the prediction is actually coming from regions which are very far from coding regions. Secondly, we see much less pleiotropy than people expected. So if you were to compare a pair of predictors, like the predictor for heart disease and the predictor for breast cancer, you would typically find that they're largely disjoint. The regions of the genome that are controlling your heart attack risk are not the regions that are controlling your breast cancer risk. And there just is much less overlap between the control knobs than what people had originally thought. The other thing that we can say on a more technical level is that the level of nonlinearity in the individual interactions between genetic variants is much less than people had originally thought. And to get into this topic more, you really have to dig into, maybe look at that review, the first review article that I indicated. Let me turn to a slightly different topic here, which is embryo genetic testing. So about 5% of all babies in developed countries today are born via IVF. In Denmark, it's actually 10% of all babies. So that means that there are millions of embryo genetic tests done per year worldwide. And the most common test is just to look for something called aneuploidy, which is to look at the chromosome structure and make sure that, for example, the embryo is not going to grow into a baby with Down syndrome. And a second most popular thing is to just screen for individual Mendelian variants like BRCA, for example. That's typically done if there's a family history. Now, using the same biopsy for which is taken in order to do those millions of genetic tests, we now can do much more than that. We can characterize the genome with great precision, and then we can compute all the polygenic risk scores and polygenic predictors that I've been describing in the previous slides. And so that allows a much more fine-grained level of embryo genetic testing than was possible just a few years ago. One technical trick that we use to get a very precise genome for the embryo, because we're just taking a sample of just a couple cells off the embryo and a small amount of DNA, is that we use the parental genotypes. We genotype the parents as well, and then we actually use the parental genotypes to do error correction on the embryo genotype. So here's an example of a report, the kind of information that you find in the report. Individual diseases are listed there. The one marked in red is aneuploid, so you wouldn't want to implant that one. The one on the bottom is an outlier for breast cancer risk. And a typical scenario, a very common scenario, is a family has a family history of, say, breast cancer, but they're not BRCA carriers, and they want to screen against breast cancer so that the next generation doesn't carry that high outlier polygenic risk. And that's a real application.
Time's up.
Oh, okay. Let me just stop then. This is the first baby born that was screened in this way, and she was born about a year ago. Her name is Aria. Her father is an MD, PhD trained in genetics. And if you want to learn